Hello, everyone. I'm more than happy to be here and to introduce and get you through our distributed delay neural mass model and explain and show some of its applications. So basically, the traditional formulations for brain modeling, they lack of some representation for time delays, specifically for the modularization. And in some of these representations, they use time delays as a fixed point in time, which is not quite realistic. And that's why we propose a distributed delay neural mass model where the time delays are represented and defined by a distribution along time. And for that, we are going to also explore the effects of brain-like dynamics and identify some key parameters that are crucial for brain dynamics. As well, we simulate some seizure-like activity and make some benchmarking with empirical EEG data, specifically for epilepsy pathology. So why delay matters in brain modeling? Let's first define what do we understand for time delays, and is the time that some neural masses need to transfer information between them. So it is being shown in the literature that time delays are crucial for oscillations and for synchrony, and which will affect, of course, the pathology transitions, and it's very important for determining and studying some seizure activities, like in the case of epilepsy. In this study, we follow the Janssen and Reed neural mass model formulation, that it contains 3 neural masses, 2 excitatory and 1 inhibitory neural mass. And the classical and traditional formulation is given by a system of differential equations, as we can see here, where it contains the state variables, the parameters of the system, and a connectivity matrix that contains all the strength connection between neural populations, as well as an external input. So this couple strengths is given or is collected in a connectivity matrix, and in our approach, we substitute this connectivity matrix by a connectivity tensor, where not only the connection stress and strength is analyzed, but also the time delays is incorporated. And in previous formulation of neural mass models, time delays have been treated as a fixed point in time or a distance-dependent function. So that's why we are also using now a distributed delay that is more realistic. This study was already published, and we were able to simulate thousands of cortical columns, and we explored the connection between them, and each cortical column could be associated to a particular brain region, so you can add some specificity to each cortical column simulation. For this numerical integration, we use the local linearization method that has been proved that is a bit more stable than traditional numerical integrators like Euler-Maruyama and Runge-Kutta, and it preserves the dynamical activity of the origin system. So basically, this numerical integrator, he used the inputs. He finds some inputs for each neural mass, and it will be treated as a known value, so makes the integration more faster. Here I am showing some of our previous results, where we make an exploration of several time delays functions, and we also try with different connectivity topologies. And as we can see here, we explore the direct delta function for time delays. That is the one commonly used. And we also analyze our distributed delay for 3 different topology metrics. And as we can see, for the direct delta function, we got an alpha rhythm. And when we incorporate our distributed delay, this rhythm shifts completely to a zeta peak. So it means that the way of interpreting and defining time delays is crucial for analyzing and defining brain dynamics. And specifically if we want to be more focused on some pathologies. So let's go now from abstract biological grounded distributed delays. So in our previous work, we only define a distributed delay framework, right? So now we are interested in incorporating more biophysical information. And for that, we are introducing some axonal properties, like, for example, the length of the axon, the diameter, and G ratio that is given for the myelination information. So this information is going to be collected in our delay tensor, as we can see here. Right? And of course, we are also considering and taking in account the connection strengths between neural population or cortical columns. So how did we extract all this biophysical information? We got these values from already published literatures. We got the axonal diameter. For that, we used the generalized extreme value function distribution that it was shown to be the better fit for axonal diameters. After that, for the G ratio, we took some data from previous work from T1 and diffusion weighted imaging, and with the G ratio and the axonal diameter information, we were able to compute the conduction velocity of the signals. So including with the conduction velocity and with the length of the axon extracted from tractography, we will finally be able to compute the distributed delays. So I'm going to show now some comparisons between our previous distributed delays, that it was more gamma-based, and this axonal-based new approach. So basically, both of them share the same delay topology, as we can see here, but that this topology can be defined as you wish. And in this case, as we can see, the gamma distributed delay contains or has smaller values than the axonal-based distributed delay that is more biophysical informed. So here I am showing some of our recent results. Is just like the time series of using both distributed delays approaches and the phase state of both of them. And as we can see here, like for the realistic axonal-based distributed delay, we are able to get a more richer attractor, and also we got a Shannon entropy higher for these axonal-based distributed delays. So in this case, both simulations have the same parameters, only these delays are changing. We made a sensitivity analysis of our model to see which parameter affects more to the brain dynamics, and to reflect how important are these axonal properties that are being included in our formulation. And we got that the excitatory ratio and the external inputs are quite relevant, right, for the seizure dynamics and steady-state dynamics, and this has been already demonstrated in previous literature. But what is interesting here is that we got that the diameter of the axon is quite relevant for the frequency of the system. So it means that a more biophysical informed distributed delay is necessary for a good representation and modeling of brain dynamics. So I'm going to show some of quick application of our model. We use our model to test this pipeline, that is a very simple pipeline for classification and anomaly detection on EEG simulations. So basically, we keep a unit, a cortical unit, and we include a thalamic circuit, which will contain an excitatory thalamic relay and inhibitory thalamic nucleus. So this will be our new unit, and we make a simulation of thousands of this entire unit to be able to simulate some epileptic or seizure-like activities. So we generate some data, and with that we train our pipeline, and after we try and we test it on empirical data. So we are seeing here some epileptic patients and healthy subjects, and we got, with the simulated data, we got around 95% of accuracy, and when using with the simulated data, we got a 95% of accuracy, and when tested on real data, we got around 89%, which is quite good. So we are trying now to make it better, and for that we are going to use some transfer learning and domain adaptation of the methods. So I want to give you some insights of our next steps. I am very interested on extending this approach to be able to model other modalities like fMRI, for example. And for that, we are planning to use the laminar fMRI model proposed by Camille. So we are going to change the Wilson and Cowan that it is mainly used in this approach, and we are going to introduce our distributed delay neuron mass model. And another ongoing collaboration is with the Aix-Marseille University, particularly with Victor Girsa's lab. So we are trying to explore how distributed delays affects the brain dynamics in the well-known epileptor model, and see if we are able to predict some of these propagation seizures. I also wanted to mention that this collaboration is funded by the MyTax Global Link Research. And well, with that, I would like to thank my PI, Alan Evans, and all the closer collaborators for your support and guidance. And yeah, that's all for my talk. Thank you so much.